Hello, and welcome to the Road to Genome podcast. I'm your host, Helen Bethel, genetic counsellor and workforce development and education lead for the North Eastern Yorkshire Genomic Medicine Service, or GMS. Today, I'm delighted to be joined by Paul Roberts, who is here to tell us more about his role within the North Eastern Yorkshire GMS. Hi, Paul.
Hi, Helen. Good to be here.
Yeah, great to see you. So can you just start then by telling us what is your current job title?
Yeah, sure. So I'm classed as a consultant scientist, and I'm also the lead scientist for solid cancer for the North Eastern Yorkshire Genomic Laboratory Hub.
Okay, that sounds really interesting.
Well, yeah, I've always been, since I came into genomics many years ago, I developed quite an early interest in cancer genomics, but at that time there wasn't that much going on in the sphere of cancer. So I started out working in all aspects of genomics. And then when we had there was a national reconfiguration of services about 6 years ago, I think it was. And our geography created a partnership with Leeds, Sheffield and Newcastle. And one of the roles that was identified there was the role of a lead scientist for solid cancer. And that was the one I really wanted. That was the role I was really keen on. And it looked like it was a great direction to take the service forward. So essentially, I have a role which, although I'm based in Leeds, it covers Leeds, Sheffield and Newcastle, the whole population, and it's a strategic role really that looks at trying to harmonise the genomic testing in solid cancer for our whole population. There's a lot of kind of political challenges there, working between 3 large laboratories, working remotely. We had COVID as well, and the whole sort of remote working, which made some life a little bit difficult. And as I said, I'm a consultant scientist as well. So within all of that, I have a service commitment and particular expertise that I've built up over the years. And that one sort of supports the other one, really. So I get a better understanding of what I'm doing on a strategic level if I'm actually at the coalface and helping out with the service work.
Yeah. So can you explain a little bit more then about what the role of a clinical scientist within cancer is or does?
Yeah, it's changed a lot over the years. We'll probably talk a bit more about that later. But at the moment, the clinical scientist is mainly involved in the actual reporting of the different tests that we do in the laboratory. So these are tests that are undertaken on the tumour samples that come specifically for genomic testing. And there are multiple different types of tests that we do. So part of the role of the clinical scientist is to understand the different tests that we do, to help to implement them, because genomics is always changing. So there's always a role to develop the tests, bring in new technologies, improve the way we do things. And then once we have new technologies embedded into service, the scientists they have to understand the role of those tests, understand the information that those tests produce, and then create reports which are easy to read for the people that refer samples to us. And this is our key objective, really, where we're producing reports, genomic reports to clinicians. So these can be oncologists, pathologists, the people that are involved in the clinical aspects of cancer care. And it's really important that we're clear what we say in terms of our results, what we found, what it means, and what the implications it might have in the context of the pathology and the other clinical information on patients.
Yeah, and that sounds like a real challenge, knowing how much the science has, almost exploded over the last decade or two, to actually be, the ones responsible for interpreting that science that is happening in the labs into something that's readable and understandable and applicable for for clinicians to do for their patients. So what a skill set you all must have. That sounds amazing. So if we can think back to your childhood then, Paul, was this the role that you wanted to do as a child? Did you always know you wanted to be a clinical scientist? Or what?
No, I was one of those kids that had lots of different ideas about what I wanted to be when I grew up. I think I started wanted to own my own pet shop when I was about 6.
Yeah.
So I'd always liked animals. I used to keep grasshoppers and beetles in my bedroom secretly. So my menagerie consisted of things that I got from my garden. Worms, I used to play with worms in the garden as well.
Fantastic. So does being a clinical scientist run in the family then?
Quite the opposite, really. My mum was a dinner lady. My dad worked in the warehouse, and for most of his life, he never really talked about his job. But my understanding was he just used to sort out nuts and bolts, put them on wagons, and they get distributed elsewhere. But he spent most of his working life talking about his 2 years national service.
Really?
So for much of my childhood, I thought my dad was a war hero. My mum was a dinner lady, and so in terms of careers guidance, they just said to me, Paul, just do whatever you want to do.
Yeah.
And school were the same. They just said, Paul, you can do anything you want to do, which is great. It's nice to have that. But then you think, well, how do I decide what I'm going to do?
Yeah, yeah. And did either of them have any interest in science at all?
Only from the point of view of animals. So
Yeah.
my mum always liked animals.
Till she discovered them living in your bedroom.
I'm not sure whether she did discover them, but she never actually let on if she did discover them. I can also remember taking a bucket full of crabs into a hotel, and I just left it in the hotel reception area. And later on, when I came down to rescue the crabs to take them back to the sea, they'd all escaped.
Oh, goodness.
So yeah, I think my childhood was punctuated by all those sort of events. Yeah. And then I moved on to, I wanted to be a sports commentator.
Okay.
So I was really keen on the whole radio aspect.
Yeah.
And I think I used to annoy my parents a little bit there, because I would, if I was playing any kind of sporting games at home, I'd be commentating on those. And that I still continue with that. I'm one of those dads who, when I'm playing football with a bunch of kids, I'll be commentating on my own performance. Sometimes I bring in the kids as well, yeah. So maybe that's why I didn't make it. I just focused on my own performance. Yeah.
Brilliant.
But as for science, no, I think it's a little bit later on through school. And my interest in animals, I got much more interested in biology when I came to high school, and the sciences. And then by the time I got to be about 17, 18, I had an idea that I wanted to do something with science.
Yeah.
An uncle of mine suggested I went into dentistry, but I had a bit of a fear of dentists. So that was a big no-no. And I couldn't really decide, so I did zoology at university, just to bide my time a little bit, and try to work out what I wanted to do then. And thinking back on any careers advice that I've had over the years, I can remember somebody coming into school to talk about being in the army, working down the pit, and being a trawlerman. So those were my three options up until I was leaving school. Yeah. And then when I went to university, similarly poor career offering, really. I was suggested I join the Hong Kong police force. Well. Yeah. So I think it was difficult to come up with a clear plan.
Yeah. With so many diverse avenues, I imagine so. Yeah.
Yeah. I mean, I was one of the first people at my school to go to university.
Okay.
As well, which I didn't really know what university was. I knew it was something that you could do just to avoid working for a short time. And it also gave you an opportunity to study something you were really interested in a bit more detail. And I think it was during that time that my interest in biology is cemented so much more. When I finished university, I thought Hong Kong police force, no, that's a big no-no. And I had a place to do a teaching course. I was going to train to be a teacher again, just to get another skill and bide some time before I made a decision. And I was all set to do my year's teacher training at Manchester University. I'd had a placement in a school. And then my mum got in touch and said, "Oh, Paul, there's this job going in Leeds. You might be interested." And it was advertised in the local newspaper, a really small advert. Yeah. So I applied for it, not knowing a great deal about it at that time. And it was a Leeds genetics lab as it was then.
Wow.
And I went in there, and I loved the idea of being able to wear a white coat, have chemicals around me, work with something that was vaguely connected to medicine. I didn't honestly know what I was letting myself in for at that time, because there was only maybe 8 people in the lab. Yeah. And it was a case of within my first couple of years at work, I've begun to get a much better understanding of what genetics actually meant. So it's chance.
Had you done much genetics or genomics in your degree? When did you first come across genetics?
Not a huge amount. As a zoologist, we used to look at Drosophila genetics.
Yeah.
That was pretty much it, really. We did a bit on Darwinism and some of the old theories of evolution. But my work at St James's was probably the first time I'd had real experience of what genetics was about, and the whole idea of being able to apply that in a medical context. I never knew anything like that was possible before.
Yeah. So really lucky that advert was spotted by your mum.
It was chance, yeah. I'd like to say I always wanted to be a geneticist, but it was chance. Yeah.
Fantastic. So thinking about your kind of clinical work, and I know you mentioned that your role now is a lot more strategic, but thinking back to your clinical work, kind of what's the typical involvement for a clinical scientist with a case? Do you get to see much of the patient, or is it a bit more distant than that?
Yeah, that's one of the abstract things about working in a hospital environment, because you're working amongst many other clinical and technical and administrative and support colleagues. And certainly when I first started, it was very much working in the lab. Samples came in. We didn't even know too much about the different clinicians that were sending referrals to us. And I think over the years, we did get to have a lot more liaison with different clinical teams, working with as I mentioned before, the pathologists, the oncologists, going to multidisciplinary team meetings. But even with all of that, even with that extra clinical contact, we still never really got to see patients. So it was very much about, we get a brief bit of a patient story. Usually it's just a little bit of details in terms of what type of cancer that patient may or may not have, perhaps what symptoms that they've got, how urgent is the testing, how old are they, and within that context, our job really is to undertake the best test that we think we need to undertake for that particular cancer type. But it is quite abstract, yeah. Even when you produce your final results, we're providing those for the clinicians who will see the patients ultimately, and will follow that patient journey, undertake the treatment and patient management. But we don't really get to hear that end part of the story, and that can be quite interesting, quite challenging, really. But I think what our motivation really is to ensure that we complete our part of the picture, knowing that further down the line there are clinical consequences. Yeah. Should we be meeting patients? Would it be nice to meet patients? I have met a number of patients through over the years. I think there's definite positives to that from time to time, just as a reality check in terms of helping us to understand the implications of what we're doing. And I did get involved with a teenage cancer project probably about 15, 20 years ago through one of the specialist nurses in Leeds, Sue Morgan. Yeah. So she was quite heavily involved in that particular unit. And there was a project two projects being undertaken there. One was a project called What Colour Is My Cancer? And this was a sort of joint genetics histopathology project where the nurses were facilitating some meetings with a number of the teenagers that have been diagnosed with cancer that have been tested in Leeds. And the idea there was that under supervision of the specialist nurses, we would meet the teenagers and we'd show them what we'd found. So we'd have microscopes set up. Wow. I took photographs over of what the results looked like, and they were able to look and see what we'd seen down the microscopes. That's incredible. And then tell us a bit about their journey, and sometimes we'd meet the parents. How did you find that experience? It's always great to have that story and that reality check for us to have a better understanding of why we're doing what we're doing. And even just to get a sense of what their understanding was, because as young people with quite vivid imaginations, they were seeing the cancer as all kinds of different things, from something equivalent to an infection, an alien that had invaded their body.
Yeah.
And even within one family, there was some sort of blame amongst the parents as well. Is it my fault that he's got cancer? And,
of course.
those are things we don't really appreciate what it actually means to the to the individuals.
Yeah.
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 And the second part of that work was there was a dance project that was started with a company in Leeds called RJC Dance, and that RJC Dance were working with the teenagers to put on a dance performance. And my daughter was 13 at the time, and she was a bit of a lively 13-year-old. So I thought it'd be quite good for her to get involved in something that was related to what I was doing, but something that would help her own development. And she and a couple of her friends who were dancers joined in with their teenagers. The idea was it was a joint teenage cancer, and then maybe other teenagers as part of that as well, and put on a performance with a dance display. And it was brilliant to see. Great for my daughter to be involved there.
Yeah.
And the fact that she was working with teenagers that had amputations, that were having treatment, that were, some of them were not that healthy, and she just saw them as other teenagers.
Yeah.
It wasn't bothered about what was wrong with them. Well, she was, but I mean, it didn't make a difference to her. To her, it was about, these are the people I'm dancing with. Let's make it work.
Yeah. Yeah. Fantastic. They sound incredible projects to be involved in.
Yeah, that was a great one to be involved in. Yeah. And I think Sue Morgan went on to do loads of other things like that.
Great. Can you tell me then, can you think of a case that has maybe stuck with you that you've worked on? So maybe a result that was maybe particularly challenging. Can you think of one that has kind of stuck with you during your time as a clinical scientist?
I think we have lots of those, really. And as I say, because we don't really meet patients, sometimes it's hard to remember individual cases as such, unless you've actually been to the multidisciplinary team meetings. Thinking about this in terms of recent examples then, so we did have a brain tumour sample came to us a couple of years ago from a youngish patient. She was about 19 or 20. And at the time, there were two different areas of the tumour. One of them looked more aggressive, more advanced than the other one, and the other part of the tumour was looking a little bit more benign. So we were sent two different samples from this particular tumour, and they both had slightly different genomic changes within them. So that our results basically indicated that the two samples had related changes. So there's the less aggressive sample had got minimal genomic changes, and the more aggressive one had got additional findings on top of that initial change. So I think the clinicians were thinking the patient's got two separate brain tumours. How did this arise? What's going on here? And what we were able to show was actually the two were related, but one had evolved from the other one and started to behave quite differently. And I think for us, it was a means of almost seeing a different kind of cancer journey. So these two things existing in parallel. Yeah. One was at the early stages, one was at the later stages. Yeah. Often when you have a cancer, you just see whatever stage it's at the beginning or at the end, whereas we're seeing sort of the beginning and the end. Yeah. If you like. And
you've seen that process in real time.
Yeah.
Yeah.
So when we talk about interesting cases for us, it's more about when we find something that's really unusual. Yeah. And we actually we're able to solve it.
Yeah. Yeah. Fantastic. And so how many years is it that you've been working as a clinical scientist then?
Well, I came here in 1982, as I said, just to get some work experience.
And you're still here.
I'm still here, I never expected to still be here.
And that's all within NEY, is it all within Leeds?
It is, yeah. I came into what was called then the cytogenetics lab. So they said there was 8 of us in that laboratory. And on a Friday afternoon, we used to sit waiting for work to come in.
Oh, really? How things have changed. Yeah.
And other scientists would do everything then. So they'd do the sample preparation, we used to culture all the samples that came in. We'd grow them in incubators, and then we'd create microscope preparations, and we'd be looking at the chromosomes down the microscope. So my role became very much about my involvement in growing the solid tumour samples as they came in. So I helped to set up that service for us. monitor how they were growing, and then when they reached a particular level of growth in the vessels we grew them in, take them out, prepare them, and then end up producing the microscope slides. And then this is how unsophisticated things were then. For us to take images of what we could see down the microscope, we had a camera rigged up to the microscope. I'd load the film in, take photos, go into the darkroom and develop them myself. My specialism was photography.
Really?
Take our own photos, and that was our output.
Yeah.
And you get lost in the darkroom sometimes. And I remember one winter's day, I'd gone into the darkroom for the afternoon to go through the whole processing. And I came out, everybody had gone home. All the lights were switched off.
And they'd forgotten about you.
Yeah, and you come out of the darkroom, you can't see anything when you've been in the dark all that time. And then even less when you come out of that.
Yeah. It never ceases to kind of amaze me when we do look back, not even that long ago to like 1982, and to think that back then the science was that we were looking at things under the microscope, like that was the detail that we could gain. And yet today, little of that work actually goes on with microscopes. It's very much about sequencing and reading those sequences, and it never ceases to amaze me how quickly that change has come about.
Yeah, it's amazing, isn't it? Especially having been in the field so long, I've seen all the different technologies in genomics come through, and what's led up to their introduction, and how we've phased out one technology and brought a new one in, and how the newer staff have come and been trained in those new developments. It's been quite exciting, really.
Yeah. And what would you say a good day looks like for you?
Well, I think most days are good days. I'm quite a positive person, really, so I look forward to coming into work. But a good day really is when everything's working. So we've got no equipment failures. The staff are in, the staff are motivated. And when I'm actually in work, a good day is when you can feel the sort of dynamic of the team. And because I think genomics is such an exciting world to be working in, knowing that we're all working there, pulling together for the same cause. And it's quite satisfying to have that. So I think a good day is when people pull together, not just in the team, but also because of the strategic work I do and the liaison that I have with different areas of our geography. When I can also sense that with the clinical meetings that I have, and that when I have the contact with my colleagues in Sheffield and Newcastle. So the good days when I can feel that teamwork, that pulling together. And it makes sense of why we're doing what we're doing, really.
Yeah, that's great to hear. And on the flip side of that, what is a more challenging day for you at work?
Well, it's the counterpart of what I've said. So you come in there and the equipment's broken. So an example of that was a few years ago, we had a big power cut in the workplace. And I was in the middle of processing a sample, and we had to do that by torchlight.
Goodness.
Yeah, not in the dark room, but so I think difficult days are when things don't quite work as they should, and because genomics is such a complex science, there's inevitably going to be times when you can't quite achieve what you need to achieve in that day. So the difficulty there is when we can't manage to fulfill our objectives for that time. It could be because there's, as I mentioned, equipment not working as well as it should do, the staff not having the stuff that we need, and I'm having then sometimes to cover and help support the team to try to get things done, because we've got that pressure put upon us to be able to turn our results around quickly, because we come at the sort of back end of the pathway. So the patient's been seen by a clinician, the sample's been taken, it generally goes through pathology. Sample then comes to us for testing, and we've got a very limited amount of time to turn those results around. So bad days can be when the pressure is on us to hit turnaround times, and when we're not achieving that.
Yeah. So can you tell us a little bit more then about your role as lead for solid cancer? What does that involve?
Yeah, I wondered what it was going to involve when I first started, to be honest, really. I think initially it was about trying to harmonise the services between Leeds, Sheffield and Newcastle, because we had three big genomics laboratories who'd all done things quite differently. And there was areas of commonality between them. So I think quite a lot of that was looking at who my main colleagues were going to be in the three different centres, who I was going to be working most closely with, how we're going to be able to share the information that we needed to coalesce what we're all doing. So there's quite a bit of my work then is working with the scientific teams to make sure we're all on the same page, and because we've been trying to consolidate a lot of the work that we do as well, and Leeds is what's called the central laboratory for our GLH, and that means more and more of the actual wet laboratory work, the sequencing work, is undertaken in Leeds. So part of the first few years was the gradual movement of the sequencing work for solid cancers, at the very least, into the Leeds laboratory. And then the way we work it is that the samples that are then sequenced in Leeds, the data that's produced from those samples goes out to my colleagues in Leeds, Sheffield, and Newcastle, to the cancer teams there, for them to analyse and report, which we call it distributed reporting. Yeah. Because I think at first there was a little bit about a fear of how that new relationship was going to work. Is Leeds going to take over the whole arrangement? But in reality, what we've tried to do is maximise our effectiveness by centralising a lot of the testing. But from the scientific team, the analytical teams, things have not really changed a great deal, because they're getting the data back from Leeds rather than from Newcastle, or rather than from Sheffield, in the same format as the Leeds scientific and analytical teams are getting as well. So that's how it works on that, in terms of the scientific teams. I keep in touch with what's happening in terms of the technical developments, because genomics is changing all the time. So I work with NHS England, and my counterparts in the other 6 GLHs in England as well. So we do share expertise. At first we were reluctant to share because we thought we were competing, but now there's very much a sharing environment.
That's great to hear.
So we can keep abreast of what changes we might need to make to try to develop and improve the technologies we're using. NHS England as well, they produce the National Cancer Test Directory, and that's a bit like that's our catalogue of testing that is commissioned by NHS England. So as new tests get added to that testing directory, we then have to make sure that within our GLH that we're able to deliver that test. If it means using the same technology and making available that test for a new type of cancer, that's fine. But sometimes it might mean working on a new technology, and that can take time to bring into play. On the clinical side, because we have to work very much with the referrers of our service, and because I'm working with solid cancer, most of the clinicians that I work with are pathologists from around the geography, and oncologists from around our patch as well. And sometimes clinical geneticists, actually, because there are some cancer types where there is an inherited component to the cancer. So there are some of the meetings where we have contact with clinical geneticists. So to improve that clinical dialogue, we've got about 8 or 9 different clinical groups that we set up over the last few years. And they meet probably 2 or 3 times, or maybe 3 or 4 times a year. And the idea behind that is they're cancer specific. So we have a clinical team that works with, say, gynecological cancers. Another one works with sarcoma samples. And the intention there is so that we can have that dialogue with a mix of the technical, bioinformatic, scientific, and clinical teams to be able to discuss the current service as it is for that cancer type, and then changes that we may need to make, or even suggestions that are coming as to how we can modify what we do and improve the service. Roughly.
It sounds fascinating. It sounds fascinating, Paul. So what do you think is the most exciting thing then coming over the horizon for genetics or genomics?
It's hard to pinpoint one thing because it's all been quite exciting, really. I think that it's following on from what's been happening in the last couple of years, really, because up until about 4 or 5 years ago, we were only testing 3 or 4 of the main cancer groups, and that's massively expanded to include the great majority of different cancer groups. And with all of that has come a big expansion in the numbers of tests that we're doing, and the types of tests we're doing. And I think the biggest change that is coming and will be coming will be how to broaden out the amount of testing that we're doing for the cancers that are coming out. So at the moment we tend to look at a very small number of genes when we look at a cancer. the challenges are going to be to look at a greater number of genes as more and more treatments are coming out that target acquired genomic changes within the cancers. So the challenges are going to be how we broaden out our service to look at a greater number of targets, but more importantly, how can we speed up the whole process? And I think one of the most exciting things that's probably going to come out, maybe by the time I've retired, or possibly even over the next few years or so, is going to be looking at much more rapid, so-called non-invasive type testing. So looking at blood samples, possibly even urine samples, to look at elements of the cancer genome that are carried around in the patient's blood.
Yeah.
And with all of that, potentially you could avoid having to work with biopsies. You could be able to have the sample much earlier rather than waiting for biopsies to be taken, and potentially be able to turn your results around very quickly and monitor. Because that's one thing with working with solid cancers, we have to wait till the biopsy's been taken. And any kind of monitoring from a genomic point of view, we have to wait for another sample to come if there is going to be another sample. Whereas with a blood sample, there's a potential there to have that broader spectrum of monitoring, a bit like what happens in leukemia. Leukemia, you can take a bone marrow sample, monitor how the patient's doing, and have a greater sense of how well they're responding to treatment. Is the disease coming back as such?
As you were talking, I was thinking of the parallels of what Polly Tally shared with us when she recorded a podcast, was that similar, kind of the fact that turnaround times are probably where most of the work is needing to come from to make sure that they are actionable in a time frame that is useful for treatment and for the patient. And similarly, that kind of circulating tumour DNA, kind of rather than, needing biopsies and what have you, as you say, which can be hugely invasive, actually you might be able to get that information from a simple blood test.
Yeah, I think so. And there is the cancer vaccine program as well that's out there at the moment that's been in the news fairly recently, looking at tailoring very much a patient-specific vaccine according to their genomic profile in the cancer. Yeah, and
fascinating.
If that comes into being, that'll be amazing, because we talk about personalised medicine, but that is very much about the individual, isn't it? It's creating something very specific to that one particular person based on their genomic profile.
Yeah. Exactly. Yeah. Incredible. Well, thank you so much, Paul, for coming to talk to us. It's been fascinating to speak to you and hear about your journey.
Thank you very much.
Thanks for joining us today. If you would like more information about the topics that we have discussed, then you can find these at our website, www.ney-genomics.org/podcasts, where you'll also find all the episodes in our podcast series. These can also be found on Spotify, Apple Podcasts, and all other podcast platforms.